been working for Illumina for 5 years, and so everything I'm saying is obviously from an Illumina perspective. And also I'll point out that this is sponsored by Genome Atlantic and the Ignite grant, and we're certainly happy to be a part of the program, so thank you. All right, so to start, I'm going to sort of introduce this, the idea of sequencing and how we can use sequencing in both research and also in healthcare from, from the beginning, from babies. Show of hands, how many people like babies? All the pediatricians kept their hands down, I think. That's how that works? No, I'm just joking. But yeah, I mean, everybody loves babies, and certainly we want to be able to have children when we want to have children, et cetera. But there are a number of cases, a number of points in the reproductive life cycle that sequencing can actually play a role. From the very beginning, so when you're deciding to have a child, if you want to know if you're a carrier for a disease, you might want to check and see if you are. If you're having difficulty getting pregnant, you can do in vitro fertilization and the like. Once pregnant, we can monitor the pregnancy and monitor the health of the child throughout. And then after pregnancy, we call it post-pregnancy, we can also call it life, everything beyond, right? After birth until death, we can monitor things throughout our lives. And so it's a little bit more granularity. I'll point out a couple of these things as introductions. So starting with carrier screening, on our MySeq DX, and I'll introduce the MySeq a little bit later in a little bit more detail, but we have a version of the MySeq which went through FDA clearance at the end of last year, so we're very happy that it got the 510K clearance. And it was submitted with 3 different tests, and 2 of them are cystic fibrosis tests. So one is a 139 variant assay, the other is a full gene test, so the cystic fibrosis full gene, and it also went through with a universal kit, so any PCR-based method that you want to use, any panel that you want to develop, you can use our universal kit, which has been FDA cleared, so you can use the FDA cleared kit with the FDA cleared sequencer. The 139 variant assay is actually really neat because even though the ACMG guidelines give you 23 variants to work with, and that encompasses most of the incidence of cystic fibrosis, that's based on typically white European descendant populations. It doesn't really take into account the variation you see in African and Asian populations, and we're seeing a lot more of that diversity come into the mainstream. So having a wider assay is much better. Also, with that particular assay has, I think, 6 of the 9 variants that are identified or treated by the Kalydeco drug. So it's actually very useful to be using that particular assay at this point in time. So moving along, if you're interested in having a child and having difficulty in vitro fertilization is often a resort that you can go to. And one of the issues with it is really maternal age. So as the mother gets older, the successful pregnancies go down, the rate of successful pregnancies go down, and it's directly related to aneuploidies of the embryos. And so the idea is if you're looking at embryos in vitro, you want to pick those that are not aneuploid. And how to do that is fairly difficult. If you look at them by eye, you can't really tell. But we have a company that we acquired last year called Bluenome. They have a microarray-based method where they actually test some of the DNA from an embryo and look for aneuploidies. And using that assay, they've increased implantation by over 60%. And now they're trying to move that entire assay onto sequencing and increase the efficacy of the assay. There's also, once you get pregnant, you want to check the child to see if it's healthy in utero, and typically we're looking at things like Down syndrome or chromosomal level aneuploids that are in utero. And if you were to do that now, you'd be doing something like amniocentesis, which carries a 0.5 to 1% risk of fetal miscarriage. And what we found over the last dozen years that there is DNA circulating within our blood that is not confined by cells. It's cell-free DNA. And the DNA is there because of apoptosis. So we have cells that naturally senesce and pop open. The DNA is released into our bloodstream and usually is quickly degraded. But in pregnant mothers, up to about 10 or 15% of that DNA is derived from the placenta, which is derived from the baby. So we can actually sequence the DNA within a mother's blood and be sequencing the child at the same time. And so this particular assay has been done in the US. There are 4 companies that sell this assay. Illumina sells it directly through Veronata, which is a company we acquired last year. Sequenome, Ariosa, and Natera all use the Illumina technology for this. So basically taking a blood draw from the mother, do some sequencing, and know with phenomenal accuracy. And there was just a paper that came out showing it's 10 times more accurate than traditional tests for determining chromosomal level aneuploidies in utero. And it's a very large market. I mean, in 2012, I don't have the numbers for last year, but about 100,000 tests ordered, largely through Sequenome. But they expect this to grow very, very rapidly. Right now, we're looking at the high-risk pregnancies, but we're going to be moving more and more towards the low-risk pregnancies because you can gain a lot of information from this sort of test. Once the child is born, things can happen, right? And how many people have seen this kid before? Just Marco, funny. This is a child, his name is Nicholas Volker. Normally we wouldn't know the kid's name. In this case we do because his entire story has been profiled in the Milwaukee Journal Sentinel. He's a kid from Wisconsin. When he was born, he had an issue with his bowel. They thought he had an odd form of Crohn's disease. By the time he was 5 years old, he had spent most of his life in the hospital and had over 200 surgeries. He removed his entire colon, and when the disease went up into his small intestine, they realized it wasn't Crohn's. They ended up sequencing his exome and found out that he had an odd form of immunodeficiency that required a bone marrow transplant. Since he's had that bone marrow transplant, he's been doing very well. In fact, you can go on, if you look for 1 in a billion and go to the Milwaukee Journal Sentinel, they're still talking about him. They're just kind of keeping updates going. So it's a great case, right? Where sequencing came in, found a causal variant, and they were able to act on that and actually treat a kid who was really on the way to death without this sort of treatment. And there are other kids like him, some of whom we know by name, some of them we don't. But there are lots of anecdotes like this where we're seeing sequencing step into people's lives and make a difference. A lot of these are rare disorders, but they don't have to be. They can be cancer, they can be other things. And it's great. We can see all this and we say, great, this is good for us. You know, we're doing what we want to do, right? This is the whole point, right? But how accurate and how good is it? And this is a paper that just came out in February that talked about exome sequencing and how many clinical diagnoses are made from clinical exomes. And what they did was they looked at 35 different studies and found that the success rate was 22.8%. Now, mind you, these are the ones that have failed every other thing. So this is 22.8% of the people that didn't have a diagnosis and weren't going to get one, that now have one. So that's a plus. But on the downside, we have over 75% of these people are missing something. So why aren't we getting all of them? How come we have gaps here? And it's a pretty big gap. And you know, part of the explanation for that is, like I mentioned, these sorts of studies are done on rare disorders, but also on not rare ones. This is an article that was in the New York Times talking about the Cancer Genome Atlas. This is a consortia that was working to profile cancer genomes, and I'll get into some of their studies in a little bit. But the take home from this was that the Cancer Genome Atlas, their charter is running out. It's completed, so they're done. And are we done? Are we done profiling cancer? No, and that's a problem. And one of the tweets that came out of this was from Dan MacArthur, who said, we need more genomes. And if we go one more step further, this is how Howard Jacobs said, this is a quote he made over a year ago at a meeting that we had. The dumbest that we are about our genome is right now. He said it a year ago, it was true then, it's true today. It continues to be true because he didn't put a timestamp on it, I'm going to keep using it. The reality is we're dumber today than we will be tomorrow. So every variant that we identify and every link that we make to a disease will speed up the analysis of the next patient that comes through and improve our understandings moving forward. And so in the end, when thinking about this, like, yes, we know a lot, and yes, we're learning a lot every single day, and tomorrow is going to be a little bit better than today, but we still need a lot to do, and we still need to sequence more genomes. And with that as a premise, we introduce the HiSeq X10. This is the largest capacity sequencing system ever imagined, ever created. It's being sold as 10 different instruments, and these are the specs on one instrument. So each one produces 1.8 terabases. I know most of you can't even fathom what a terabase is, but what that comes out to is about 16 genomes in 3 days. So if you can contrast that with the original Human Genome Project, which took 15 years, it cost $3 billion, we can do 16 in 3 days, or 5 a day, at a cost of about $1,000 each. So speeding things up and doing this at massive scale. Who's doing this? People who are interested in studying island nations. All right, anybody here own an island nation that they want to sequence? Because if you do, we can talk about the X10. Honestly, these are the types of projects that we want to be a part of because they create great value, right? The Faroe Islands has been trying to sequence their entire population. It's about 60,000 people that live there, so they can identify genetic signatures of the Faroe Islands. Same thing in Iceland. Decode Genetics is now one of the customers of the X10. And in England, at the end of 2012, they announced that they were going to sequence 100,000 people over the next 5 years. And if you can see, if you know anything about sequencing, This is a gel-based technology that we stopped using a long time ago, so it was really, really far behind. We got to help them out. All right. In fact, we've sold the X10 to a number of different places. This is all of them that I know of at this point in time. I don't think we planned on selling this many, but the interest in this particular platform has been so grand that we've sold 9 of them at this point. Each one of these, I should point out, since it's 10 systems, cost at $10 million. So if you have $10 million and an island nation, give me a call. All right. But not everybody is interested in sequencing whole genomes. There's a lot of data that's generated. It's a lot to do to analyze that amount of information. And most people are studying parts of the genome, so like exome studies. And this is an article that came out from the Cancer Genome Atlas. And the question that they were asking, or at least that I'm going to point out here, is how many exomes do you need? How many samples do you need to understand what you're looking at? And in this case, they were looking at 5,000 tumor pairs, and they were looking for the variants that they could identify within those exomes. And in red are the most common variants. So these are the ones that occur in 20% or more of the samples that they looked at. And it looks like they kind of max out around 1,000, and then you kind of get diminishing returns as you add more samples. So about 1,000 to get the most common variants. But every other level below 20%, so if you look at the orange line, this is the 10 to 20% variants, maybe we're starting to plateau there at about 5,000 samples. 5 to 10%, we're clearly still in exponential phase, and these other guys, we're really just at the bottom of the curve. So the driving home the same point, we need more. We need more information. Another thing that these guys did to kind of maybe give you a little more positive take on this is they profiled different tumors. And looking at, in this case, they're looking at the mutational burden. So on the left-hand side, with the lowest mutational burden of the 12 types that they were looking at was AML. And on the right-hand side, with most is the lung cancers, which makes sense because the lung cancers are dealing with, actively dealing with mutagens, whereas AML is not. So that kind of makes sense. And then you point out what type of changes, C to T, C to G, et cetera, the different variations that can occur and how they're different between the tumor types. And it's interesting. It's more academic, but it's interesting. Where it becomes a little bit more useful is something like this, is a heat map of the types of mutations in the genes that were affected. And so we start off over here with P53. I know these letters are really, really small and you can't see them, but this one right here is P53. And I've known, I'm sure it's been common knowledge for many years, about 50% of all tumors have a P53 mutation, right? What I didn't know is that it's variable. So 95% of ovarian cancers that they looked at had a P53 variant in it, whereas only 2.2% of the AML samples that they looked at. So it is different between different cancers. And so, okay, so P53 is mutated in many of them, more so in some than others. But what about if you are interested in AML? Here's a gene called NPM1 that's mutated in 27% of AMLs, but really not in any of the others. So maybe you want to look at the pathways that are affected by NPM1 and not so much worrying about P53. And this is a way that you can now focus efforts on the pathways and the genes that are more relevant for the cancer that you're looking at. I think this is kind of neat. So I've spent hours kind of looking at this because it's kind of cool to kind of parse this out in different ways. But I'm a geek, so what are you going to do? All right. For these sorts of studies, we have a different system. It's called the 2500, the HiC 2500. It generates about 1 gigabase of data, so it's about half the throughput of the X10. About 4 billion DNA molecules we sequence at a time, and the read links are we sequence 125 in one direction and 125 in the other. So it can sequence a great deal of information. Putting this in human genome context, you can do about 10 genomes in about 6 days. So it's a little bit less data than the X10, but it's still a lot of information. Again, thinking back to the original human genome, 1 in every 15 years, here we're doing 10 in 6 days. So it's quite a lot. It's an interesting system because it has 2 modes. It has the high output mode that I was just talking about, the 1 terabase. But it also has what we call a rapid run mode. In this case, the flow cell's a little bit different, so it has 2 lanes, not 8, and so it can turn around the data much, much quicker. A 2 by 125, same read length on this side would take about 20 hours. So you're talking about an overnight run. So you can do one human genome in 1 day and get that data back very quickly. We're going to be extending the read lengths out in a couple of months. So it'll go to 2 by 250. This is useful for a number of different applications when you're trying to rebuild genomes, and also for certain types of analyses. The longer the reads, the better. And so we're going to be doing that in rapid run mode. And unlike the X10, which we kind of limit to human genome because we're really focused on that with the X10 system, with the 2500 you can do anything you want in terms of sequencing. Whether that's, you can do human genomes if you want to, you can do exomes, you can do RNA sequencing to look at transcript profiles. I put ChIP-seq in here, but it's not limited to that. You can look at methylation, you can look at microbial sequencing. There's pretty much anything that anything you can consider that has DNA or RNA in it, you can sequence on the 2500. Now, at the end of last year, Illumina had essentially two instruments. We had a MiSeq, which has a small throughput, and I'll talk about that in a little bit, about 15 to 20 gigabase of output, whereas the 2500 was looking at 600 gigabase, and in between that's a huge hole. And we were told that pretty much every day. Hey guys, you're going to put something in the middle here. We have a little one and we have a big one. We have a little one and a big one. And now we have something in the middle, and we call it the NextSeq. Essentially what we've done is we took that rapid run mode that was in the HiSeq 2500 and put it into its own box to make a really easy to use, simple to run device that's powerful enough to run a human genome at a time. Okay. So a couple of things about the NextSeq. We announced this just a couple of weeks ago, so I'm going to go in a little bit more detail about what it is. So it looks just like this, and it has a touch screen, so it's really simple to run. You just actually touch the monitor, click the button here that says sequence, and it'll instruct you what to do next, etc. The specs on the instrument, it can produce from 20 to 120 gigabase. Again, 120 gigabase is about one genome. 20 gigabase is small panel sizes. The number of reads that we can use, this is the number of DNA molecules that we can sequence, goes from about 100 million to 400 million. And the quality and the read lengths are very good. This means we can sequence 150 bases in 2 directions. Okay. And all of this makes it very convenient, and also the price makes it convenient as well, since it's about a third of the price of the HiSeq, which in the US it's listing at $250,000. So the HiSeq 2500 is $740,000. So the couple things that make the NextSeq really convenient are the speed, flexibility, and ease of use. The way we got to the speed, the way our sequencers work, we start off with a library that looks like this. So the yellow part is the fragment of DNA you want to sequence. We put adapters on either end. You take that library, you put it into our flow cells. They bind to the surface of the flow cells. They amplify right there on the site. And then we introduce nucleotides and sequence away. Now, the NextSeq does everything that all the other instruments do, so you can use the same libraries that you've been running on the other instruments. You can also run on the NextSeq. Everything's the same. The one thing that has changed is that rather than putting a different color dye on each of the 4 bases, we only use 2 dyes, or 2-channel sequencing. So we have a red dye associated with the C base, a green dye associated with the T base. The A base has a mixture of green and red, and then the G base is unlabeled. So when we take 2 pictures, we can say, here's the cluster. This is called a cluster. When we see it, we say, all right, that one is red only. We know it's a C. We see another one that's green only. We know it's a T. We see one that's both red and green. We know it's an A. And if we know that it's unlabeled, then we know it's a G. Okay, so very simple. And what that does for us, it means we have to take half the number of pictures. Rather than taking 4 pictures for each of the 4 bases, we only have to take 2. And so we can speed up the processing and get through to the data a lot faster. And so the way this works essentially is if you're going to do the maximum amount of sequencing that we can do, 300 cycles, it'll take about 30 hours. That would be the longest run that we have. Half that, 2 by 75, takes about 18 hours, and short runs, 75 cycles, give you about 12-hour runs. So you can do this basically in a day. In terms of the flexibility is really built into the flow cells. So we have 2 different flow cell options that we call the mid-output and the high output. You can see that the lanes here, this sort of light gray part, there are 4 of them, 1, 2, 3, and 4. And then on the high output, there are 4, and they're much fatter. And so in each of these, we can sequence. So this flow cell can do 130 million clusters or DNA molecules, and this one can do 400 million. So it gives you a different amount of output that you can generate. And on the sides here, talking about what sort of samples you can be looking at, whether it's a couple of exomes. So if you're doing 3 exomes, that's like a trio analysis, mother, father, child. Same thing with RNA sequencing. Certainly smaller panels, you can do many smaller panels. You can do a genome in the high plex, or obviously more exome or RNA-seq, and certainly other types of panels that may be a little bit deeper in depth that you might want to look at. And then for the ease of use, if you're considering running one of these, it is truly simple to run. We've had these out in the field as demos. What happens when you buy the reagents from us, they come 3 parts come. The buffer cartridge comes, the chemistry cartridge comes, and the flow cell. There's also a waste container, and they basically just slide into place. So the waste goes in there, the buffer goes in there, the chemistry goes in there, and the flow cell actually drops in the top. And then you press the buttons and off you go to load or run. To give you a better view of what these cartridges look like, all these things come pre-filled, so they're ready to run, except for this one right here, the one with the orange around it that says load your sample here. So that's the one thing you've got to do. We got everything set, ready to go, but we need the DNA or RNA. Once you put that in there, you can put it in the box and we can sequence it. And that's basically all you need to do. The data that's come out of R&D, early returns on this has been very good. So using Phi-X as a little virus, it's probably the most sequenced thing ever because we use it as a control. So we've sequenced this little 25 kb virus many times, but in this case, 2 by 150, 120 gigabase of output, over 400 million reads. The quality scores were very good. And here you can see what the error rates are, and they're all below 1%, which is very good for that read length for us. This is a different sort of output, but this is a human sample, and in this case what we got were over 75% of the bases were Q30 or above. What that means is that we predict that any base that's Q30 means one base out of 1,000 is incorrect. So a very high accurate, very good system. If you're interested in looking at RNA transcript profiling, this is just showing the difference between the NextSeq and the other instruments that we sell. So the HiSeq is over here and the MiSeq is over there. So if you're looking, so HiSeq on this side, MiSeq on this side, and then the NextSeq is the comparison. So when you're looking at RNA, you want to know if certain genes are upregulated or downregulated. And if you take the same sample and sequence it on both instruments, you should see basically X equals Y. A straight line, and that's essentially what we see. And the R squared here to define that line is 0.99, 0.99, 0.99, 0.98. So we're getting very good tight coordination between the different instruments. What's even more interesting is when we actually plot this, and what we're looking at is the same gene, this is GAPDH, sequenced on each of the three systems, and each one of these is a sort of a read pileup. So every time we sequence something and it lands on in one part of the genome, in this case it's RNA, so it's going to land only on exons. We want to know how many times does it land right here, and the higher the amplitude, the more reads we get landing there. Okay? And what you can see is that all these little curves are exactly the same on all three systems. So there's not even like a change in the coverage. We're getting exactly the same information on all of the systems. So we have 3 different ways to sequence. You get the same thing each time. We do also get a lot of questions about our unlabeled G. When we don't label the G, oftentimes we get asked, how can you sequence GC-rich regions? And the answer is very well. The way our sequencers work, we identify where the cluster is at the beginning of the run, and then we sequence one base at a time after that. So it doesn't actually matter if we can see it or not. We know it's supposed to be there. It's sort of like if you closed your eyes and turned the light off, we know that you're all still sitting here. Once we identify where you are. And the same thing is true with our sequencers. So with GC-rich regions, this is 70% GC, or huge GC, which is typically about 85% GC. We can sequence them as well as with our other systems. HiSeq uses four dyes, so we can see all 4 bases. So basically, it doesn't matter whether we can see the G or not. We can sequence GC-rich regions very well. Oftentimes, people ask about pricing. So there are 5 different reagent cartridges that are sold with the NextSeq, and this is the list prices for each of those. So we have for the mid output and the high output. So basically the way our systems work is you buy a reagent cartridge, and one run will cost you these amounts, and then you can toggle your per sample prices by putting more or less samples in there. So if you put 10 samples in this run, they come out at $97 each or something like that. That makes sense. Another way to look at it is the cost per gigabase, and obviously the X10 has been priced so that we can get very good pricing on genomes, so $7 per gigabase. The 2500 currently is looking at $43 a gigabase. With the upgrade to the 1T, which is coming in a couple of weeks, it'll get down to about 29 gigabase, and the X10 is really well positioned to compete with the 2500. So your per run costs are very similar to the bigger system. Okay? Now, I mentioned before the Cancer Genome Atlas and these monstrous studies that they're doing in cancer, and I didn't want to scare anybody by just showing those. I want to show you that there are also small studies in cancer that can be extremely valuable and can be run on an instrument like the NextSeq. This started as an N of 1, where a single individual had a brain cancer. They pulled out several mets, and they ended up looking at a bunch of people to confirm what they saw, but what they found was a gene fusion. And they found this by looking at both the transcriptome and the exome. And they found that NAB2 and STAT6 were actually merged together, and in doing so, it was causing the cancer. Really great paper. They did a number of other studies. They looked at gene expression levels on arrays to coordinate with the RNA-seq that they saw. They also made a NAB2 STAT6 gene fusion, transformed it into cell lines, showed that those cell lines grew faster, and were able to put together the model in which NAB2, which is a transcriptional repressor, when fused to STAT6, becomes an activator of the genes it normally represses. And that in and of itself is enough to cause cancer. So pretty cool paper. And this is, again, a small enough project where it could be done in an XSeq and you don't need a full consortium of help to get that done. All right, moving on to the MySeq. The MySeq's are our smallest sequencer. It has a lot of the same easy-to-use features that I already talked about on the NextSeq, the touch screen, like easy-to-load cartridges and all that kind of stuff. One of the areas that well, a couple other things about the MySeq that make it even easier. It has a data server inside, so it has a number of outputs that it can generate. So it doesn't just do the sequencing, it also does data analysis. And one of the cool things about the MySeq that we don't talk enough about is how flexible it is. So it has a number of different flow cells that can give you from 1 million DNA molecule reads all the way up to 25 million. So about 25-fold variation in the amount of data that you generate. We also have different read lengths associated with those, so we can go from 50 bases all the way up to 600. So all told, you can generate from 300 megabases all the way up to 15 gigabases. Kind of a wide range of the amount of data that you can generate. The data server, like I mentioned, has a number of different applications, from standard resequencing, where you're just aligning your reads to a known reference. You can do de novo assemblies if you don't know what you started with and you want to build the genome, you can do that. There are, again, a number of different things that can be done here. I'm not going to go through all of them. Another way of looking at the different applications that people like to use. I'm going to talk for a few minutes about metagenomics. I started my life as a microbiologist, so I like talking about microbes. There have been a number of projects that have been going on in the world in the last couple of years, all of which take sequencing into account. Have you guys heard about the American Gut Project or the Microbiome Project? I've got a couple nods there, that's good. This is where they're crowdfunded, so you send them some money and they will send you a collection tube, then you swab yourself, and then you send it back. And then they'll sequence it and they'll give you a report saying what microbes are growing in or on you, depending on where you swabbed. It's kind of very cool. I mean, last summer they had a program where they had people enlist, basically, and go on a diet. So they check your gut microbiota at the beginning of the diet, then you'd go on a diet for 60 days, and then they check you at the end, and they wanted to see how your gut microbiota changed with a particular diet. It was kind of funny. I mean, they made people eat like 30 different kinds of vegetables every week. You know, my son would be right out. Not doing that. But you had to sign up, and if you did it, you'd have to do things like, you know, if you're eating broccoli, you have to eat the florets, but also the stem, right? You can't wimp out on the stem, and things like that. But I think you could do this yourself, right? You can sign up and get 2 collection tubes and go on whatever diet you like, right? You can make up your own, you know, beer and pancakes, or, you know, that would be fun. And then wait 60 days or something and check it again and see how it goes. Anyway, you can do this, and you're contributing to their scientific efforts as well. So there's a lot going on there. Another project, this is a friend of mine, his name is Chris Mason, he's at Weill Cornell. He's doing what they call the Pathomap project, where he sent his students and postdocs out in New York City to swab every subway station. And if you were in New York City and you saw somebody swabbing, you probably looked twice. But they were there and they were checking the microbes there. I let Chris know that I was talking about it, so he sent me slides. This is a data slide that he generated. So there's lots of stuff that they were finding in the subways, a little bit scary. But what's interesting is that some of this was indicative, right? So in this case, they were able to identify that this was on the turnstile and they knew exactly where it was. These guys are found everywhere. This guy was found on a bench. So you can identify not only, you know, every location has a unique identifier. And so you can see the changes over time, which is kind of funny. And he imagined something like, you know, nowadays we get Amber Alerts, or we can get Amber Alerts on our phones, but he imagines a day where we will get a pathogenic microbe alert, and you'd find out, you know, if you're going to Grand Central Station, you know, you better make sure you're washing up. I think you should be washing up anyway, especially if you look at the list a little bit more carefully about what's actually there already. But anyway, it's a clever idea, and I think Chris is also thinking about expanding his horizons beyond New York City. And so if you want to start the Halifax Genome Project, you can go ahead and contact Chris. I'm sure he'll incorporate it into his project. But I saw this cartoon and I thought it was definitely him. I don't know if you can read this in the back, but it says, "Excuse me, I'm looking for the dirtiest possible thing I can touch on this street." So yeah, that would be him right there. All right. There has also been some uptick with government entities. So the FDA in the U.S. has purchased 85 MySeqs for foodborne pathogen surveillance. Public Health England and the CDC in China are both doing epidemiology studies, so any outbreaks that occur, they'll be able to be on the scene very quickly and be able to sequence the microbes very fast. Been showing this for a while, but not until last week was I able to show the impact of this. In the U.S., just a couple of weeks ago, there was a place, Roost Foods, that was shut down as a conclusion of an FDA investigation of a listeria outbreak that was associated with some cheeses that they made. And this was a case where they found 8 people were infected, and 1 of them died. And they were comparing PFGE, the standard method for typing bacteria, with whole genome sequencing, and they got a clearer distinction. And this is from the CDC. Their report on this said they had a clearer distinction with the genome sequencing versus PFGE. So this is, actually seeing the fruits of the investigation and the whole project that they put forth. We were actually seeing things happen, so this is great. And luckily, only 8 people were affected. All right, but it's not just, actually, before I get into that, there's one other story I would tell. At AGBT this last year, did anybody go to AGBT? It's a meeting in Florida. A guy named Joe DeRisi told a story in which a hospital contacted him about a child who was in the hospital with encephalitis. Now, there's about 100 different things that can cause encephalitis. They were having difficulties figuring out which one it was. There were some complications also about the individual who had autoimmune issues. So they were having difficulties figuring out what to do with the boy. He ended up in such dire straits that they put him in a medically induced coma. And it wasn't sure what was going to happen to him, but they were able to take a sample from him. They gave it to Joe DeRisi, who has a MySeq, and they also took some of that sample and sent it to the CDC. And the CDC was going to run qPCR, their standard tests, to figure out what was going on, and Joe did sequencing. 2 days later, they had data back from the MySeq, which identified leptospira as the causative agent. They figured out how to treat the kid. 2 days later, he was walking out of the hospital and going home. The CDC, about a week later, reported back using the qPCR study that it was inconclusive. And I think that's ultimately where we're talking about this, right? The ability to use something that has a much greater clarity, a better understanding about what's actually there, is the better tool. And the MySeq is the better tool than qPCR. All right. Now, it's not just about microbes. On the MySeq, we're also doing panels. So looking at human genes, but looking at fewer of them. We're not looking at whole human exome. We're not looking at transcriptome. We're just picking a subset of those. And we have panels like the cancer panel, which has 94 genes in it, including BRCA1 and BRCA2. Sometimes we call it the Angelina Jolie panel, right? We also have cardiomyopathy and autism panels, which also have about 100 genes in them. The inherited disease panel is a little bit bigger. It's got about 500 genes in it. The tumor panel, this is not for predisposition. This is if you've got a tumor, and now you want to sequence that, and it's meant for FFPE tissues, et cetera, and looking specifically within a tumor to find out what's driving that tumor. The most interesting one to me anyway is the TrueSight1 panel. This is a much larger panel that encompasses 4,800 genes, 12 megabase of target. And the idea behind this is that you can use this panel if you're running a clinical lab. Validations are difficult, but if you have one panel that you can validate for one panel, you can actually filter the data back, and you can report back the autism panel, report back the cardiomyopathy panel, or any other panel that you choose to make up. But that's all informatic. Basically, in practice, you're just validating one panel, and you're getting all the genes that are relevant. These 4,800 genes are all the ones that are known to be associated with disease or are drug targets. So despite the fact that we're sequencing exomes, oftentimes when you look at the data, it all ends up falling into the same 4,800 genes. And the selection was pulled out of OMIM and other databases, so we know which genes are typically associated. And this particular panel can be run on any of our sequencers, so on a MySeq, the NextSeq, or 36 samples per lane on a HiSeq. And then we also have methods to help you analyze that data, so it comes back quickly and easy to interpret. One of the tools that comes with this is called Variant Studio. So once the data is generated, we get all the sequences, align it, find variants, we get what we call a variant call file. That becomes input for Variant Studio. The Variant Studio will annotate those, so it'll tell you which genes associated with it. It will translate them, and it'll tell you, all right, that mutation is going to be it's in the wobble position, it's benign, it doesn't mean anything. This mutation actually is a stop codon that has known pathogenicity, and here are references to support that. And so this Variant Studio tool is really, really great, and it'll not only classify things, but it'll go right into generating a report. So basically, you can take the variants and go directly to a report within a couple of minutes, and then generate the report and pass that off to whatever the next step is. In addition, we acquired a company called NextBio, which is essentially a data mining company. I don't know if they want me to call them that, but what they do is you put in a search term into their engine, and they'll report back everything that's possibly known or publicly available in all databases that they can have access to. And that's going to be sort of the engine driving Variant Studio and a lot of these tools going forward. Currently, it's not. So I'm kind of curious to see how that's going to be implemented, but very excited to see NextBio's power being built into the Variant Studio tool. All right, a little bit more practical. This is a study that came out of Bert Vogelstein's lab down at Johns Hopkins. In this case, the question was about ovarian and cervical cancers. And these are problematic in that they're typically asymptomatic until late stage, and then they're difficult to treat. And so what do we do to try to find the earlier stage? We do pap smears. And so they were asking, can we use a pap smear and sequence and get better, better diagnostic power than what we typically do, which is histological or PCR-based, or really looking at certain things? Can we do better than that with sequencing? So what they did was they started off with a study where they were looking at 22 tumors and also normals, starting with a pap smear. So they extracted the nucleotides from the pap smear itself, and then identified a number of genes. There were 12 genes, 50 amplicons in total, that were indicative of cancer and not present in the healthy samples. And then they translated that into the MiSeq and into a test they're calling the PAP gene test. I don't know where it is in clinical development at this point, but I think that's the idea, to move it to a clinical test. But when they tested this, as a proof of principle, they got 100% sensitivity and 100% specificity. So all the tumors came up tumors, and all the normals came up normals, just using 12 genes and 50 amplicons. And running this on the MiSeq is a quick turnaround. It's a one-day test. So pretty cool to see that sort of the research hopefully moving towards the clinic. All right. So one of the things that we often get asked about is preparing the sample. So for us, sample prep is taking nucleic acid, putting adapters on it, and getting it ready to sequence. I know for many of you guys, sample prep is getting it out of a cell or out of blood or something like that. So what we want to do is, we'll call it template prep. Is that what was I supposed to call it? Template prep? So I'm not going to call it sample prep, we're going to template prep, and make it as easy as possible. So once you've gotten to the nucleic acid part, can we make this simple? And one of the things that we're going to be introducing this summer is called NeoPrep, and this is a microfluidic system. So basically, you take your nucleic acids, you put it into a cartridge, press go, and it will create the library ready to sequence for you. It uses microfluidics, and this is a method called electrowetting. So each of these gold bars is a conductor that you apply voltage to, and when you do that, this droplet, which is sitting, it's a water droplet with some filler oil around it. If you apply a voltage there, it'll move on top of it, and if you then move the one next to it, it'll move on to that one. So basically, you can move droplets around and generate things. So I'm going to try to show you a video that's on our website. I tried to put it into the talk, but it didn't seem to work. But hopefully, this will work. No, it's going slow. We liked it the first time.
Imagine library prep with far fewer manual steps.
Oh. Harsh. All right. Well, we'll skip that. Basically, the idea is that the cartridge moves the little droplets around. You can see them a little bit here on this. On this image here, you can see the droplets here, and they basically move through these tracks very, very well, and basically you don't have to intervene at all. You put your sample in, it moves everything around, does the sample prep for you. You come back later and you're ready to sequence. Crushing that didn't work. All right. So over the course of the last hour, I've told you about all of our different sequencers from, population scale island nation projects on the X10 to the 2500, which can do exomes, RNA sequencing, as well as genomes. The NextSeq, really easy to use, very flexible, can do small projects, large projects as well. The MySeq, microbial work, as well as gene panels, and even the DX, which is doing carrier screening and CF testing as well. So a lot of different things that can be done. Along the way, I've told you about a number of different areas that we're doing research, reproductive health, infectious disease, etc. Ag, I didn't talk about ag. I did mention broccoli. Maybe that counts. All right, but there's a number of things going on there. I didn't talk about forensics, which is also a really cool area that we're moving into. So a lot of stuff going on. And I thought I'll just say thank you and take any questions that you have, and hopefully address any of your pressing needs. Thanks.
Thank you. If one's objective is to run routine targeted panels, is there any downside to having the NextSeq as opposed to the MySeq?
The MySeq does some data analysis for you. The NextSeq does not. That would be the downside. So basically, when the data comes off, you're going to have to put it someplace where you can do the data analysis. So that would be the one caveat.
Okay, in terms of cost of running and that sort of thing?
The cost per run is going to be substantially cheaper on the NextSeq.
The next.
Yep. You can do more samples at a time. You have, again, multiple flow cell options. So if you don't have that many samples, use a smaller flow cell option, and you'll still be able to get a much more cost-effective sequencing run.
Thank you.
Yep. I've scared you all.
Just to follow up on that question, you mentioned that the NextSeq does not do the data analysis. So when would you have to buy another platform which you could then do the data analysis?
There are a couple of things you can do. For time purposes, I pulled out some of the informatics solution slides that I had in here. We have a method that we call BaseSpace, which is a way for you to port your data into the Amazon cloud. And from there are applications to do the data analysis for you. I know in hospital settings, porting to a cloud is not going to happen, I know. But we also have a version of BaseSpace, which we call BaseSpace Onsite, which is a analysis server that we can sell with the NextSeq for doing very simple to use. It's basically point and click data analysis without having to learn any command line stuff. But if you didn't like that, then any kind of server would work. All right, well, thank you everybody for coming, and if you want to talk privately, well, Marco and I is here. I didn't introduce Marco. Marco Raposo is our clinical specialist, so we'd love to chat with you. Thank you.